All right, I think I'm going to get us started with a bit of introduction, and we'll see if anyone else is able to join us today. So, hi everyone, my name is Leon Gua. I'm the research coordinator for California Sea Grant, standing in today for Tanya. And today we have our fourth and final webinar of our month-long weekly series highlighting our 2022 Graduate Research Fellowship projects. And I just shared in the chat our webinar series flyer, but I will share it again once I finish talking and giving this overview. So for those of you who may not be familiar, California Sea Grant is a federally funded and university affiliated organization that supports applied science in ocean and coastal fields for all of California through research grants, fellowships, extension programming, and communications and outreach. The Graduate Research Fellowship is one type of our many fellowship opportunities that our program provides, including other research, policy, and extension fellowships. And you can visit our website to learn more about these other opportunities that we have. And again, I'll drop those links in the chat where you can also sign up for our newsletter. We started the Graduate Research Fellowship Program in 2020 to directly support graduate students working on applied research that meet our strategic plan focus areas of creating resilient coastal communities and economies, sustainable fisheries, and healthy coastal ecosystems. During the 2-year fellowship, fellows are directly mentored by a research mentor and a community mentor, and they develop an outreach plan as part of their project to ensure that their research is useful for the entities and communities invested in their work. And so throughout June, we've been really proud to feature talks by our 2022 cohort of graduate research fellows sharing all the great work they have accomplished over the past 2 years. So today we'll be starting with Rachael Ryan from UC Berkeley, whose talk is titled Impacts of Increasing Climate Variability on Coho Salmon at the Southern End of Their Range. So I'll just give a minute for Rachael to get loaded up here, and I'll share some of those links I mentioned in the chat. So right now, Rachael, I see the whole PowerPoint screen.
Is that better?
Yes, it's going through like an animation, but I think it is loading up. So, all right,
Okay.
go
Awesome.
ahead. I'll spotlight you and we'll get you going.
Okay, great. Thank you. Thanks everyone for tuning in. My name is Rachel. I'm soon to be finished PhD at UC Berkeley, and I was fortunate to be a fellow the last 2 years working on spawning in endangered coho salmon. So, there we go. Just a little bit about me and my background. I started snorkeling for fish back in undergrad at McGill University in Montreal, Quebec. And I fell in love with streams and fish, and it led me to a lot of beautiful places around Eastern Canada. But I came to Berkeley to work with Stephanie Carlson and Ted Grantham on salmon because I was really fascinated by their life history diversity, as well as the challenges that they're facing, especially in California, in wake of climate change, especially with drought. And it was during the extreme drought of 2021 where I was in the field and observing a lot of juvenile salmon mortality and kind of these ecological traps in streams that usually are perennial. And so I got my advisors and I talking about kind of the spawning restrictions on spawning during drought that are kind of this blueprint for juvenile life history. And so it kind of dictates where they are in the landscape and how fish are able to grow. So we kind of zoomed in on this spawning aspect, especially spawning in relationship to rainfall. So if you're familiar with California coastal streams, you know that the flow is really low in the summer, and it really waits until it requires that winter rainfall to elevate it so that fish, especially salmon, can then ascend streams. Also, the rainfall magnitude and frequency really dictates as well kind of these cues for spawning salmon. However, as I'm sure you're all aware, our precipitation regimes are really shifting, and we're starting to see these effects already in delayed onsets and kind of shorter and sharper precipitation seasons. So this was a really good time for us to start asking questions about precipitation variability and how that impacts spawning distributions. And we did this in Lagunitas Creek, California, which is in Marin County. It's a small watershed, about 270 square kilometers, and half of that watershed is inaccessible to salmon because of dams. However, it's actually quite a diverse small watershed. So in the colors here, I have purple, which is the Lagunitas Creek subbasin. It's the largest subbasin of the watershed. And this main stem here is regulated by flow releases from the reservoir. And then in the blue here is Lima Creek, which is an unregulated tributary, and it has some intermittent tributaries to it. And so there's actually a high level of geological diversity, and topography, versus topography and vegetation that lead us to being a pretty unique watershed. In addition, it's a stronghold for endangered California coastal salmon, coho salmon, and this is one of kind of the key populations that's still wild origin. And so not only is it really important to study its persistence, but also it kind of gives us a really interesting window into how these wild salmon are moving across the watershed as adults in space and time. And so, as I mentioned, this watershed is divided in kind of these regulated subbasin and natural flow subbasin. And the way that the regulated subbasin is operated by Marin Water, the municipal agency, it is for the benefit of salmon at different life stages. So in the fall, for example, it elevates winter base flow around November 15th if there's no storms. It can also provide pulse flows. It can decrease variability. And so it's actually really beneficial, regulated in a beneficial way for salmon. And so this actually came in a time in 2021 where this regulation was starting to be questioned, or at least reconsidered, because in 2021 we were seeing the reservoirs were getting really low in terms of their water sources. So this was an opportune time for us to look at how this flow regulation is influencing interacting with spawning, because this was going up for debate. And so our project really is looking at patterns of spawning across the watershed and looking at precipitation variability and how it influences those patterns. So our partners, some of our collaborative partners, Marin Water, as I mentioned, they both operate the reservoirs across the watershed and also do a lot of the life cycle monitoring in Lagunitas subbasin, and National Park Service does the life cycle monitoring in the Lima subbasin. And so Marin Water had this concern about the flow regulation and whether or not it's actually beneficial for salmon spawning. And as that's coming up for debate, how they can kind of add science to that. And then in the National Park Service, they were actually concerned that the flow regulation was attracting some of the Lima Creek subpopulation into Lagunitas Creek and influencing and depressing their returns. And so there is this question of dispersal that they were worried about. So in collaboration with Rachel Johnson's lab at the Center for Watershed Sciences and NOAA, we kind of came up with these two main objectives. One was to characterize adult spawn timing in relationship to hydrology, and the other was to characterize dispersal. So I'll just kind of talk briefly about each one of these. So for timing, we looked at the number of reds and the onset of that timing. So when the number of reds first started, or when fish were first starting to lay eggs, and then peak spawn timing, where the majority of fish were coming up to spawn. And we looked at whether these were different between the streams and also whether the drivers were different. So those hydrological drivers I already talked about, which is the timing of the wet season, and then metrics of flow variability and magnitude. We used the monitoring data from our collaborators from 18 years, and we used generalized linear models to try and assess these differences. And because it's a short timing, I'm only going to kind of go into a subset of this in the drought, but this did encompass an 18-year time series. So during the drought of 2012 to 2016, this is where, we really would see the differences in management. And we do see how the onset of spawning in Lagunitas Creek, for example, remains pretty steady and is also sooner versus in Alima Creek, where especially in 2012 and 2014, we see it's really delayed. And so clearly there's those impacts of that delayed precipitation. And then peak spawn timing actually was conserved between the streams. And so what this means when we dive into the drivers of these trends, we see that peak spawn timing, so fish are really dependent upon winter storms. So it really still is that precipitation cues that seem to be driving the majority of spawning across the watershed. However, the onset of spawning is really delayed, can be delayed by rainfall, variable rainfall in Alima, but not in Lagunitas. And so this suggests that the flow regulation of that earlier flow elevation and decreased variability is having a positive effect and buffering against these drought years of delayed rains. However, the majority of spawning is still dependent on rainfall events. And so we still see a lot of effects of drought on spawning, but this potential tool in our toolbox of ecological flow management. When we look at where fish who are spawning are coming from, we can dig into their origins using otoliths. So otoliths are calcium carbonate earstones that are in fish, and they grow proportionally to fish. You can think of them as kind of like tree rings. And what's most important in this context is that they can incorporate the chemistry of the water around them. So streams have kind of a chemical fingerprint, and the colors here can kind of indicate their unique fingerprint in this stream. And otoliths, when you polish them and identify their natal origin, you can get the fingerprint of the stream that the fish was in during that natal rearing period. What that means is that you can then take your returns that have died in a certain stream and spawned there and find their natal origin to assess dispersal. I'll just show you, we did 10 years of this, but I'll just show you kind of the first few or some key ones, I guess, to show you the trends. And what we found was that in Lagunitas Creek, fish were mainly coming from Lagunitas. And so this is a pretty self-sustaining subpopulation. We didn't see any Alima fish coming into Lagunitas. So that shows that that flow regulation is not attracting those fish. However, in Alima, we did see that there were some of these Lima fish coming back, as well as we were able to isolate this intermittent stream here and found that in some years, especially, this intermittent stream had outsized contributions to the adult population. So highlighting the role of intermittent streams in adult returns. We also saw Lagunitas fish coming back and some fish from outside of the watershed. So a Lima population really is this mix of fish that's being subsidized from different origins. So this dispersal through space. And this is particularly important when we have really intense drought years. So in 2012, when we saw that really delayed spawning, we actually did not find any three-year-old Alima fish return. And so suggests that maybe that cohort could have been wiped out, yet we have dispersal from other areas and dispersal from fish through time, where we have jacks that only spent 1 year in the ocean and they're coming from different cohorts. And so this highlights that our maintaining habitat diversity as well as age structure, so life history diversity, for example, can support returning adults and contribute to this population and its persistence. And just to bring this all full circle, when I talked about that 2021 drought that I witnessed this year in 2024, I went to the stream and I saw fish that had returned and it had a pretty good spawning year. And we saw fish that are 3 years old, but also a lot of 2-year-olds and even one 4-year-old. So, and they were recolonizing some of the streams that had gone dry in 2021. And so this shows that we look at this population as kind of a meta population that's able to be recolonized. Then if we maintain, use our toolbox to kind of use ecological flow management and maintain and restore habitat diversity, we can really contribute to adult returns in the population persistence. And so with that, I'll leave it with, I will be excited to keep going on some of this work on endangered salmon in response to drought at my postdoc position at Davis this fall. And I want to thank Sea Grant for the funding of this project, and I can take any questions.
That's awesome. Thanks so much, Rachel, and congratulations on the postdoc. Folks, we welcome you to add any questions in the Q&A function. And we do have a question in the chat. I don't know if there'd be any impact, but did you see any correlation between the spawning time of year, location success, and the location or intensity of the blob between 2013 and 2018?
Yeah, that's a great question. I actually have not looked at any correlation between the blob and these timing. What was interesting was that Lagunitas Creek was really consistent. So there wasn't really any change in at least the onset of timing during those years. So it does suggest that some fish are coming up. However, it would be interesting to look at the peak, so the majority of fish in correlation with those really warm marine years to see if that kind of impacted, the peak timing, especially trying to tease out kind of hydrological conditions in the freshwater with the marine conditions. So I haven't done that, but that's really cool.
Wait and see if there's any other questions, but I have one while we're waiting. I'm curious if while you were in the field, because obviously like fisheries people always meet people when they're in the field, but if you talked to any local community members and like got a sense of what their perception was of the local populations and what was going on with them.
Yeah, it's always really fun to talk to people in the field. I got a really wide range of responses. So especially in 2021, when it was really dry, I had people who have been working, volunteering. There's a lot of volunteering efforts for restoration in the Lagunitas Creek watershed. And so I talked to a lot of people who felt really disheartened, unfortunately, because they'd been volunteering for a really long time, and felt like the watershed hadn't really been improving and salmon were constantly struggling. So that was kind of a common narrative that I heard, especially when there was a restoration project in 2021 at one of the public golf courses, former golf course space, that really needed high flows to kind of engage in processes of restoration. And it became pretty, I think, an ugly eyesore for a lot of people. So yeah, there was a lot of community sentiment at that time of feeling like the salmon were doing really poorly. But then you go there when they're spawning and people are out and everywhere and super excited. So it is really interesting. It's like exactly what people can see, I think changes kind of their sentiments around how they're doing.
That's why there's so many bird people and not many fish people.
Yeah.
In the general public. I'm pro fish, so
yeah.
I know my bias there. All right. Thanks so much, Rachel. I
Thank
don't see any other
you.
questions, so I think we're going to move on to Rachelle. So Rachelle Tallman is coming to us from University of California, Davis, and their project was titled A Release Study Assessing the Survival of Juvenile Spring Chinook Salmon in the Upper Klamath River Basin to Inform Reintroduction. So Rachelle, are you able to screen share?
Yeah, let me double check. Okay, and then okay, can you guys see my PowerPoint?
Right now it's in presenter mode.
Oh, okay. Sorry, let's see here. Okay. Oh,
I think it's just taking a moment to load up.
yep, okay.
There we go. All
Sorry.
right, go ahead.
All right, hi everyone. My name's Rachelle. I'm a graduate student at UC Davis, and today I'll be talking about some really cool work that I got to do in the Upper Klamath River Basin involving spring Chinook salmon. We call them chials, so that's the indigenous word that the Klamath tribes use to describe these fish. Okay. So a little bit about me is I grew up in Florida, but I moved back and forth between Florida and California my whole life. But I've always cared about the ocean. I always loved working with animals. And I always thought I was going to become like this dolphin trainer for SeaWorld. Luckily for me, that did not happen. I ended up going to school at UC Santa Cruz, where I got an internship with the NOAA Southwest Fisheries Science Center, working on their coho program in Scott Creek. So after I graduated, I ended up working in a couple different areas with different salmoniid species. So I worked with coho in Santa Rosa, then moved up to Humboldt to work with coho and Chinook. I worked up in the Siskiyou Mountains doing fire restoration, involved that would promote salmon restoration. And then finally, about 6 years ago, I ended up in the Sacramento Valley working with steelhead. But really what ended up happening was while I was doing all these jobs, I realized that I really wanted to get back into research. But one of the biggest projects that I had worked on was in the Siskiyou Mountains involving the Klamath River. And so it was the Klamath that I decided that I wanted to dedicate part of my dissertation work to. And I was lucky enough that through Sea Grant, I was able to do this really awesome telemetry study that took place in Southern Oregon, but has humongous impacts for Northern California. So getting into the study, this is the Klamath River Basin. It is undergoing the largest dam removal project in U.S. history. Its headwaters are within this rectangle called Upper Klamath Lake. So it starts in Southern Oregon, winds its way through Northern California before entering the Pacific Ocean. The two different colors of gray that you see here represent the lower and Upper Klamath River Basin. So for any of you who have heard of this dam removal project, a lot of it is focused on the lower basin because that's where the big dam removals are actually taking place. So the dams that are being removed are Iron Gate Dam, Copco 1 and Copco 2 dams, and J.C. Boyle. However, part of the dam removal involves reintroducing spring salmon into the upper basin. So when these dams were built over 100 years ago, it basically blocked off access to spring Chinook, and they haven't been seen in the upper basin since. Basically, whatever remains of these populations are in the lower Klamath Basin, in the Salmon River and the Trinity River. So one of the big restoration goals with this dam removal is to try and create a self-sustaining population of spring Chinook. But one of the big concerns is that even though we can reintroduce these fish into the upper basin, there are still two dams that they'll have to overcome in order to make it to the ocean. These two dams include Link River Dam and Keno Dam, which are not slated for removal. So one of the biggest things that Oregon Department of Fish and Wildlife and California Department of Fish and Wildlife are interested in was, can fish make it from the upper basin through these two dams and out to the Pacific Ocean? So my study really focused in on, can they even make it from the upper basin? Because for the last 100 years, this basin has transformed into a huge agricultural area. There's been a lot of water diversions, and so there's a lot of concern that these fish might not even leave Upper Klamath Lake to begin with. But before we get into my study, I always like to give people a brief history about the upper basin. So this is the indigenous homeland of the Klamath tribes, the Yahoo-Skin Paiute, and Modoc nations. Between 1800 and 1900 is when we see European colonization of the area. And then beginning between 1900 and 1966, we actually see the rise of these big dams, most of which are hydroelectric facilities. During this time, we also see the removal of indigenous rights. Beginning in 1954, we have the Klamath Termination Act, which basically was the federal government removing the reservation lands and also not recognizing the Klamath tribes as a sovereign nation. It wasn't until 1986 that the federal government re-recognized them as a sovereign nation, but the reservation lands were not returned. At the same time, there were other issues going on in the basin. While today's talk is about salmon, there are two fish species that are a huge concern in the upper basin. They are the short-nosed and lost river suckers, both of which can only be found in this part of, of the Klamath River. So they didn't get listed for ESA until 1988, but it was actually in 1986 that the tribes stopped harvesting them because they were the first ones to notice that these fish populations were crashing. And unfortunately, in 1993, that is the last successful sucker recruitment. And so there's a lot of work going on in the upper basin currently to try to save these species from extinction because they live on average about 30 years. Getting into the 2000s, in 2002, we have the really famous Klamath salmon fish kill. So what this was there was a lot of really warm water being sent down to the Klamath that resulted in a lot of parasites, and a lot of fish ended up dying. And so there were a lot of community members that talk about this event because it was a huge moment for many of them in terms of being advocates for the river. In 2010, there was a lot of movement towards trying to bring these dams down. So 2 agreements were drawn up, but only one of them was signed. So the Klamath Hydroelectric Settlement Agreement was signed, which basically stated that the major hydroelectric facilities in the lower basin were going to be removed. The Klamath Basin Restoration Agreement was not signed, and unfortunately, that was an agreement that was going to designate a lot more money towards the restoration in the upper basin. However, some good news occurred in the 2020s. So as part of an experimental release, we released some of the first spring Chinook into the upper basin in the spring of 2022. Last summer, Copco 2 Dam was fully removed, and then this summer, we have ongoing removal of JC Boyle, Copco 1, and Iron Gate. So this kind of leads to my research question, which is, we know that these dams are coming down, and we want to create a self-sustaining population of spring Chinook, but what is their outmigration survival in the upper basin? And we chose to focus on 3 key areas: the Williamson River, the Wood River, and Link River Dam. So what's interesting about the Williamson and Wood River is that both of them are tributaries to Upper Klamath Lake, and these are spring-fed systems, which means their headwaters are springs, they stay relatively cool all year long, and this is very clear water. Link River Dam was of interest because it's their first major barrier for spring Chinook to get out of the basin. And although there is a fish ladder in place, we weren't sure if the juvenile salmon could actually migrate past this dam. So here we are again back to our study map, focusing mostly on where this rectangle is. You can see the Wood and Williamson River here as tributaries to Upper Klamath Lake, and then Link River Dam at the bottom here at the terminus of Upper Klamath Lake. So zooming in, this is actually where our study took place. And what we did was an acoustic telemetry study where we deployed a bunch of acoustic receivers throughout the Wood and Williamson, as well as upstream and downstream of Link River Dam. And the way that we did this is we did them in single or dual arrays. And what that means is receivers could either be deployed as a single line or a single receiver spanning the width of the river, or in some cases, we were able to put 2 lines spanning a river at key locations. So in this case, those key locations were at the mouth of the Wood, the mouth of the Williamson, and then upstream and downstream of Link River Dam. And the reason we did this is because we wanted to know directionality at these key points. Basically, we were interested to see, are we seeing fish going upstream and downstream at these key points? It also allows us to get better detection efficiency, meaning our ability to detect fish at these points was a lot better than some of our other ones. So the way that it worked was we went in and we tagged our fish with acoustic transmitters. Once these fish are released, they swim past our acoustic receivers, and inside is the transmitter. The transmitters emit a unique identification code that is pulsed out into the water. And our acoustic receiver has something at the top called a hydrophone, which actively listens for these unique detections. Now, depending on what kind of telemetry system you're using, some are more sensitive than others to sound. And in our case, we use JSAT, which allows us to tag smaller fish, but it also means that we get a lot of environmental noise. So anything from wind to propellers on boats can generate enough noise that our acoustic receivers will actually record that sound. So once we download these receivers, we actually have to go back in and filter the data for all of what we call false detections. Acoustic tagging took place from March 28th through April 1st of 2022. It took a lot of collaborators to do that. So each station had a tagger, a data recorder, and then a fish runner. And what happened was that these fish would then be separated into these 3 pens, either for the Wood release, the Williamson release, or we had a third option, which were a subset of fish that we were going to take to Oregon State University to be housed for future research. These fish were basically held to see, like, are we seeing issues with the transmitters? Is there any issues with our taggers? We wanted to account for anything that might impact our survival estimates. So once we completed tagging, fish were held for at least 36 hours to recover from surgery. 390 fish were released into the Williamson River, while 395 were released into the Wood River, and an additional 85 were transported to Oregon State University. The reason our release numbers weren't exact is it turns out we were really bad at counting, and so we were off by 5. In the photos that you see here, these are members from the Klamath tribes putting back some of the first spring Chinook into their ancestral river of the Williamson. So once we were done collecting data, which basically took place, we removed the receivers at the end of September. We had to go back in and remove all those false detections. And when I say that these receivers are super sensitive to noise, I had over 1 billion detections that I had to go through and filter out all the wrong tag codes because of environmental noise. By the end of it, then I would get a subset of data that I could then use to plug into a model. So the model we used is a multi-state model. In this particular scenario, it's more of like a Cormorant Jolly Seaver model. So if you've done market capture studies, it works the same exact way. Just for us, we had 2 separate releases, which meant 2 distinct migration paths until the fish hit the lake. I also like to call this model magic because it took several weeks for me to get it to converge. But basically what it was doing was it was looking at all my detections, and at each receiver location, it was estimating what the survival estimate was for that receiver array, as well as the detection efficiency. So what that means is some receivers were better at detecting fish than others, and we wanted to be able to account for that in our survival estimate. So some preliminary results. Basically, what most people I was working with wanted to know is, what is the survival of spring Chinook through the upper basin? And focusing on our 3 key points of interest at the mouth of the Wood, the mouth of the Williamson, and then upstream and downstream of Link River Dam. At the mouth of the Wood, we had 75.6% survival. What this means is, of the fish that were released into the Wood River, at least 75.6% made it to the mouth of the Wood to enter Upper Klamath Lake. On the Williamson, 74.4% of fish were detected at the mouth, so that meant 74.4% of those were able to make it to the lake. However, once we got to upstream of Link River Dam, our survival estimates dropped a lot. For the Wood River release, they were about 6.4%, and for the Williamson, this was about 9.7%. One of the reasons we see this huge drop is Upper Klamath Lake is a super shallow and wide lake, which makes it extremely difficult to use acoustic telemetry in. So ideally, you would place your receivers close by to each other to try to see the drop-off of where mortality occurs. But in this case, it was too difficult for us to try to get enough receivers to span the lake that we had to call the lake its own reach. So it's not too unusual that the numbers would drop a lot, but it definitely got us interested in understanding what might be going on in the lake that could contribute to such low numbers. However, one thing that was kind of interesting about our data was the reach between upstream and downstream of Link River Dam. So initially, we weren't sure if the fish can make it past this dam. Again, there's a fish ladder in place, but none of it was created in mind for juvenile passage. What we found was, of the number of fish that were released into the wood, 87.2% of those fish were detected downstream of the dam, while for the Williamson, 93.9% of those fish were detected downstream. And this was really cool for us because, again, we didn't actually expect them to do very well with this dam. And the other issue is, where our downstream receiver wasn't actually right up against the dam. We had to place it further downstream for safety reasons. And so there was a considerable distance from the dam to our receiver, which was also kind of cool to see, like, despite this distance, these fish are still making it downstream. But again, this issue comes up of, okay, well, you had a lot, or of those that made it to the dam, most of them made it over, but what happened to all the rest? Well, what happened was in the summer of 2022, my colleague Mark Hereford at Oregon Department of Fish and Wildlife got a bunch of calls from fishermen. And basically what they were saying was, hey, we caught your spring Chinook salmon. And at first people might be like, well, how do they know it was yours? Well, because of the dams, there have been no spring Chinook in over 100 years, which means every single spring Chinook that was caught was either released by me at UC Davis or through Oregon Department of Fish and Wildlife. And so that got us really interested in thinking about, well, maybe that estimate at the upstream Link River Dam portion doesn't necessarily reflect survival, but maybe it's more presence, meaning a lot of the fish may have not chosen to leave the upper basin and instead choosing to remain in the upper basin within cold water refugia. So part of the upper basin houses a population of red band trout, which during the summer, the lake can turn really poor, meaning there's low dissolved oxygen and it's really warm. And these red band trout will go into cold water refuge spots over summer and then come back into the lake once conditions are more favorable. And it was something we had thought about when we released these fish, that maybe the spring Chinook might do the same thing. And it turns out, because of these fishermen, we suspect that there might be a decent population that is trying to use this life history strategy. So one of the things that we did the following year was actually move a lot of our acoustic receivers away from the Wooden Williamson and instead placing them into Pelican Bay, where we could get an idea of how many fish were actually utilizing this habitat. I'm hoping in the next couple months I can go through and actually get a better idea of the proportion of fish that choose this cold water habitat versus exiting the upper basin. One of the biggest limitations, though, with working with the cold water refugia spots is that Pelican Bay is not the only place that has cold water. In the northern portion of Upper Klamath Lake, where the Wood River enters, that is actually known as Agency Lake, and that entire area is also known as a cold water refuge. At the mouth of the Williamson and Wood, those are also cold water refuges. And off to the eastern side of the lake, there are multiple small springs that these fish can hold over in the summer. So it can make it kind of difficult to figure out where these fish are going if they are choosing to use cold water refuge. However, I know that is something that researchers are interested in. And so going forward, one of the things that the agencies are interested in is looking at estimating the summer use of cold water habitats by spring Chinook. I'm hoping in the next couple months to actually work up the data I have to get a better idea of, at least in Pelican Bay, what the proportion of fish are using for Pelican Bay. For future studies, they should look at deploying acoustic receivers within other cold water areas in the upper basin, as well as, if it's available, deploying more acoustic receivers within Upper Klamath Lake. So I recently spoke to a researcher at USGS, and that is something that they will be doing this summer, is that they have 56 acoustic receivers that they are planning to span the entire lake to get a better idea of what's going on with spring Chinook in the lake. And finally, the biggest one is once these dams come down, estimating survival to not only the California border, but also to the Pacific Ocean. I want to give a special thanks to all these organizations because they provided so much funding and support to make this happen. And a special shout out to Mark Hereford from ODFW. He was the guy in the boots on the ground to help me get this done. As far as future directions, I just recently took a job with California Department of Fish and Wildlife to work on the North Coast Salmon Project, which focuses on restoring watersheds that are coho priority watersheds. And then I'd like to end my talk with a land acknowledgement for the communities of the Upper Basin. And with that, that's my talk.
Thank you, Rachelle. I think we are unfortunately out of time for questions, but I encourage anyone who does have a question for Rachelle to add it to the Q&A feature. And if we have time at the end, we will get to those questions, or maybe Rachelle, you can also answer them in the chat or the Q&A. So thank you. Congrats on the job. Very exciting. And we're going to go ahead and move on to Marissa Bills, who had come from California Polytechnic State University at San Luis Obispo. The project is titled Rebuilding an Icon, Developing Novel Approaches for Improving Management and Fostering Community Engagement to Recover Pismo Clams in California. Please take it away, Marissa.
Awesome. Thank you, everybody. Hi, I'm Marissa. And like was just said, I'm a master's student at Cal Poly San Luis Obispo. Let's see, there we go. So a little bit about me. I did my undergrad at UC Santa Barbara. And then after undergrad, I took a handful of years to just work in the field and get additional experience. And I had some really cool opportunities to work a whole bunch of different places, and mostly around shellfish. And after a handful of years doing that, went back for my master's at Cal Poly SLO, where I am now thinking about Pismo clams. Well, let's see. Okay. So if you are unfamiliar, Pismo clams are a large species of clam. They're an intertidal clam found on wide sandy beaches from Monterey Bay, California, down to Baja California, Mexico. They supported iconic declined in the 1980s. So in California, Pismo Beach was kind of the epicenter of this fishery. So if you haven't been to Pismo Beach before, we're very, very into these clams here. There's clam statues, there's clam murals. They're a very big deal. So the city of Pismo Beach was definitely impacted by the decline of this fishery. So much so that about 10 years ago, they approached Cal Poly, and they were kind of just like, what can you do to bring these clams back? And I think what the city of Pismo Beach had probably envisioned was we were just going to grow a bunch of them at Cal Poly, throw them back on the beach, and then suddenly have a fishery again. But obviously it's not that simple. But my advisor here was like, well, that's a really interesting question. Let's try to understand what happened to this species locally. So he started a long-term monitoring project in 2015 that I've been leading for the past 4 years. And one of the main components of this is doing surveys on Pismo Beach. So we survey 3 sites along the northernmost part of Pismo Beach, and we survey one of these sites monthly, doing transect surveys to collect clam abundance and size structure to try to understand just what's going on with these clams right now. So with about 10 years of data, we're starting to get a little bit of an idea of what's going on. So I'm going to show you just first the first 6 years of our time series. So on this figure here, we're looking at the mean number of clams per transect per year. And you can see that within the first 6 years of our time series, the population of clams is increasing. And we had 2 really good years of recruitment here in 2015 and 2019. If I add the next 3 years, you'll see that our figure changes just a little bit. We had a shocking recruitment happen in 2021. So my first summer as a grad student, which has been a huge blessing and a curse. It was a very exciting summer seeing just this huge abundance of clams return to the beach, which was really exciting. As one of my undergrads says, we love to see it. We hate to count it. It has made our fieldwork very, very interesting, but it's really exciting to see just such a huge recovery in this population in such a short period of time. And I'd also like to point out that this is a natural recovery and that no management intervention preceded this in any way. This is truly just the clams recovering on their own on our local beaches. So it's a fantastic time to study them. So I feel very lucky about that. And kind of with this increased abundance, we can start asking all kinds of new questions about the clams and their recovery. So despite the fact that these clams are just literally right under your feet in the sand on the beach, we know shockingly little about them. Because they are a small scale recreational fishery, they are fairly understudied, and the management agencies such as CDFW don't really have the time and resources to devote as much attention to a species like this, where the impact is fairly small. So working with my community mentors at CDFW and state parks, kind of all of the information that we can start generating about this recovery and the species is just really helpful to them in trying to understand the species better and hopefully leading to better management of the species as they hopefully continue to recover. So the kind of two objectives that I sought to tackle with my project is trying to understand their spatial distribution, their movement, and their habitat associations. So basically trying to get at where on the beach are they and why. And then secondly, estimating their growth and mortality rates in multiple ways. That's a really important metric for fisheries management. And now that I have this amazing cohort of small clams that I've effectively gotten to watch grow up for the last couple of years, we have a ton of really great data to try to get at these things. So as you can imagine, with the sheer number of clams that we've been seeing, we survey about 2 miles of Pismo Beach, which is a very labor-intensive effort. However, the entirety of Pismo Beach is about 17 miles long. So it would just be truly impossible to survey the entirety of the beach as intensively as we survey our normal study sites. But it's important for us when thinking about this entire population to know what's going on with these clams along the rest of the beach. So we needed a different way to kind of assess what areas might be suitable for clams and in what abundance. So previous work in our lab found that sediment grain size was a really good predictor of where Pismo clams would be present or absent throughout the state of California. And so I wanted to explore this relationship on a finer scale along the length of Pismo Beach. So what we did is we did paired sediment surveys and clam surveys at our 3 regular survey sites. And then we added 2 additional sites further down the southern end of Pismo Beach. And the person that you see in the bottom right corner is Tyler. He is a student who worked on this project and did all of the sediment gradation. And he definitely got more than he bargained for with his senior project, looking at more sand than he knew was possible. So big shout out to Tyler for doing the sediment work. So looking at this figure first, this is looking at the median grain size. So the sediment surveys that we did, we determined the median grain size at all of these 5 locations. And so what we see here is that our 3 standard sites, the sediment grain size is pretty comparable, but then as we move further south along the length of Pismo Beach, the sediment grain size jumps up significantly. So as we move from north to south, the sediment grain size increases. And when we add the clam data on top of that, we see this really nice pattern that as the sediment grain size increases, the clam abundance decreases. So this is really helpful, again, in that it's a lot easier to measure sediment than it is to actually go out here and survey for clams. So this helps us understand as the sediment grain size changes along the length of Pismo Beach that we can expect to see the clam population change. But additionally, if we were to think about the rest of the state, if populations like on Pismo Beach can just kind of come back on their own, this could be possible throughout the state. So if we're evaluating if there's suitable habitat throughout the state of where Pismo clams might return after their populations have declined, we could look at sediment grain size instead of doing intensive surveying for the clams as kind of a baseline metric of where they might return. So that was really exciting to see. So then again, thinking about kind of in their suitable habitat, what they're doing within the intertidal zone. So when we're doing our surveys, we divide each of our transects up into 3-meter sections, and we take a GPS point at each section along our transects. So we're effectively mapping our transects in real time. And what this allows us to do is basically assign a general location to all of the clams that we're finding. so we can get kind of their position on the beach. So to be able to make that position comparable over time, we use the NOAA National Shoreline, which is just a shapefile that we imported into R. And we use this as kind of a reference point. So we compared the clam locations to this NOAA shoreline to give each clam in our data set sort of a relative location so we could look at how their position on the beach changes over time. And so what we're looking at here is all of the clams for 1 year of data at our 3 field sites. So what I'd like you to notice with this y-axis is that it's inverted. So the top of the y-axis is higher up on the beach, so corresponding to our 0 point or that NOAA shoreline. And then as you move south or down the y-axis, you're moving lower into the intertidal. So looking at just 1 year of data, we do see very clear seasonal movement of where the clams are within the intertidal. Most notably, we see that they're much higher up on the beach in the winter months and then much lower throughout the rest of the year. And we see that this pattern holds across all 3 of our field sites. So very clear seasonal movement over the course of 1 year. And since we see this pattern repeated at all 3 field sites, we're just going to look at 1 field site over a longer time period next. So this is looking at just our medium-sized clams. So clams between 30 millimeters and 70 millimeters, again, looking at kind of their movement over a longer time period. And we see basically that same pattern. So they are higher up on the beach in the winter and then the spring, and then lower down on the beach in the summer and the fall. However, if we look at the largest size class of clams, or clams that are 80 millimeters or above, we actually see that these clams aren't moving all that much. So there's some movement, but it's really within a much smaller band of the beach. So this large size class of clams are kind of choosing to stay put and exhibiting a different movement behavior than the smaller clams. So we're not exactly sure why they're moving in general or why this movement is different based on size class. But these are things that we're trying to look into a little bit more and better understand. But it is really interesting that we're kind of seeing these movement patterns and this difference based on size class. So lots of other species in the sandy beach exhibit movement patterns, whether they're moving daily with the tides or just staying put at a certain level. So it's kind of interesting that we're seeing this species in the intertidal exhibiting a different movement pattern than some other species. So definitely something we're trying to understand more, but pretty cool to see. Again, in the interest of time, I'm not going to go too much in depth into estimating our growth and mortality rates, but to try to understand these things better, we have conducted tank experiments, a mark and recapture experiment, and a cohort analysis to try to start estimating growth and mortality rates. Again, these are important fisheries metrics, and as we're seeing all of these small clams kind of getting larger every single year, it's going to be really important to try to understand when they're going to be hitting the legal size for the recreational fishery and what that's going to mean for kind of recreational fishing. Definitely something the CDFW is very interested in trying to understand. Additionally, we've had a lot of broader impacts with this project. Probably one of my favorite things about all of this project is just the opportunity for undergraduate engagement. So I've gotten to mentor 31 undergrads in our lab kind of associated with these projects, 8 of whom who have gone on to do senior projects, either doing small pieces of this work or related projects tackling other questions related to Pismo clams. I've gotten to take about 1,000 undergrad volunteers out to the beach to do these surveys with us. These surveys would not be possible without the undergrads at Cal Poly giving us their time on Saturdays and Sundays, trying to collect all this data. So very, very appreciative to them. We've gotten to do some really neat outreach with high school students, done a lot of community events and public talks just to kind of chat with people, make them aware about these clams, they're coming back, the fishing regulations. We worked with a biology and film student who's a double major to make like a short little film about this project. And we do SciComm on TikTok and Instagram. So if you want to see us embarrass ourselves, check that out. And then again, just thank you so much to all of the funders and undergrads who have made this project possible. It is a gigantic team effort, and I'm so appreciative to all of these folks. So I'll take any questions you might have now.
Thanks, Marissa. Those clams are so cute, and I love all your cutouts.
Thank you.
Okay, there's a question for you. If you have a link for the short film, can you share it with us? would we be able to post it on California Sea Grant's website or social media?
Yes, absolutely. I will absolutely send that to you.
Thank you. And then there was another question, when will Sea Grant application open for fishermen? And I don't know if that's something that you can really speak to, Marissa. It might be related to our fisherman's apprenticeship program, in which case you should email us separately about that. Yeah, maybe I missed something. Did you say something in your talk that was related to that?
I don't think so.
Okay. I'll ask them to email us. All right, I have a question for you quickly, so I don't see other ones coming in. So you mentioned like the sediment size with the clams and their abundance. So I was curious if at Pismo Beach or in the area nearby, if they do like beach nourishment, if they add sand to the beach, and if so, how are they thinking about taking into consideration what your findings are with regards to like maintaining clam habitat?
Yeah, so on Pismo Beach, we don't do any nourishment, but this does have applications with beach nourishment because obviously in other beaches in Southern California, they are nourishing those beaches where Pismo clams have historically been. So this is definitely an application of like, there's very clearly a sediment grain size that the clams are looking to be in. It's probably, again, not just sediment grain size. It's sediment grain size is definitely correlated with like wave energy and beach slope. But sediment grain size is the easiest one of those three metrics to really kind of measure quickly and easily. But we're definitely assuming that the sediment grain size does matter to a pretty significant degree. So for those beaches in Southern California where they are nourishing, it's definitely something that they should be thinking about. And it would be really cool to kind of see how that may or may not affect clam recovery.
Very cool. Is that like a goal to reintroduce Pismos farther down the coast?
There is a group in Orange County that is very interested in that. So definitely something that they would need to take into consideration. I think originally that was probably the city of Pismo Beach's goal is like, let's just throw a bunch back and try that, see if it works. So there's definitely interest, I think, because they were just such a popular recreational fishery. I think there are definitely kind of small groups that would love to see that happen, but there's a lot of like environmental and habitat factors to take into consideration of like, is this appropriate area to try a restoration effort?
Yeah, makes sense. Great. Well, thank you so much. And thank you to all of our speakers who joined us today. Thanks for your questions as well. This has been wonderful to see what all of our fellows have been working on. And this was our last symposium. So we're really excited to hear that all of you are moving on to greater things. Marissa actually has joined the California Sea Grant team as a staff research associate. So everyone on today's call got hired. That's awesome. So congratulations. Thanks everyone for joining. We appreciate you and have a lovely rest of your day.